Hello, my name is Laura Johnston, and I'm the Scientific Associate Director of the Flow Core at UChicago. This is the fourth video in my Flow Basics 2.0 series. And again, my goal for this series is to break down the practical steps of running a flow cytometry experiment and help you understand how each of these steps ultimately impacts the final results. So for this video, I will be going over experimental design, which is mainly going over the controls, when to use those controls, what they actually do, and how to divide your cells between all of these controls. So let's get into it with the different types of controls. As you can see, there are quite a few. I've kind of grouped them into 4 different categories. So we have our single stain controls, which I would say are the most important. They're used to set up voltages on the instrument and calculate compensation. Then we have our gating controls, which help analyze the data. The staining controls help you troubleshoot your staining protocol. And then there's a few other controls that we'll talk about at the end. So for your single stain controls, which some people also call your compensation controls, those are hopefully pretty self-explanatory. They are stained with just one single fluorophore. That means that if you have a 6-color panel, then you would have an additional 6 tubes for each of those fluorophores within the panel. Usually the first question I get is, what cells do I use for those single stains? Ideally, you would want to use the same cells that are being used for your sample, your full stain tubes, but it doesn't necessarily have to be exactly the same cell type. You could use a different cell type that, as long as it still expresses the marker, or you can also use compensation beads. And I'll discuss in a few slides when you might want to switch the cell type and other factors you need to think about. Now for staining your controls, I do want to keep reminding you that pipetting is a source of error, so please do not try to pipette very small volumes. Hopefully, based on my previous videos, I've convinced you that you need to titrate your antibodies, and that may mean that some of your antibodies require very, very small volumes. And in order to pipette those for your single stain controls, I strongly recommend making a dilution. I like to make a 1:10 dilution and pipette 10 times as much, so it's much more accurate. I know it sounds tedious, but you will get better results for your controls if you follow this. Also keep in mind that you might need to manufacture your controls in order to get good staining. I will, in a few slides, talk about all of the rules that you need to follow for your controls. So in order to follow those rules, you might need to manufacture your controls. Maybe you need to stimulate your cells. Not everyone necessarily manufactures every single control, but the one that I would recommend most people create is the control for your viability dye. So if you are lucky enough to have a really good sample prep that doesn't have very many dead cells in it, then I would strongly recommend killing some cells and using those, or mixing dead cells and live cells, to create the control for your viability dye. Then you're going to know that you have a really nice population of positively stained dead cells within that control. Another note I want to point out, if you are using cells that have fluorescent proteins in them, so if you have GFP mice or GFP cells, keep in mind that those cells cannot be used for your single stains, because if you use those cells, you will end up with 2 fluorophores per tube instead of 1 fluorophore per tube. So make sure you don't have any fluorescent proteins or any other type of fluorophore within your control tubes. So as I mentioned, the single stain controls have 2 purposes. The first one is to set the voltages or the gains on the cytometer. The second one is to calculate the compensation, or if you're doing spectral, calculate the spectral unmixing. So for setting the voltage, this is a little experiment that I ran. I used the exact same single stain control. I ran it at 8 different voltages. I combined all those files so that I could show them in the same plot. And so you can see as you increase the voltage, then our positive population gets brighter and brighter, and we also start seeing more spreading of our negative population. Now, if you remember from my previous video where I talked about antibody titration, you'll find that you get a similar pattern of staining if you change your antibody concentration. So you can imagine that you want to optimize both the antibody concentration and the voltage setting on the instrument in order to get your best separation of your positive and negative populations. The good news is that you don't have to do a lot of work, hopefully, for setting voltages on the instruments. We at the U Chicago Core have done a lot of these voltration tests ourselves. So for most of our instruments, you'll have a recommended starting value, and then you would just want to use your single stain controls to make sure that you don't end up in this scenario where your sample is too bright and saturating the detector. So mostly you're going to be utilizing your single stain controls to calculate compensation. So this is an example of what you might see on the instrument. If you set up all of these plots, you'll see where the spillover is on the instrument for every color. So here's an example where I've stained a tube with Percy Psi 5.5. So this is the detector that's assigned to that fluorophore. It captures the largest percentage of the emission spectra. We see a nice population here. And then I have 3 other detectors, and because we know that we only put 1 fluorophore in the tube, we can see that Percy Psi 5.5 does not spill into the FTSE detector, but it spills into these other 2 detectors. Therefore, calculating the compensation will remove this spillover. Now, another way to look at it, again, this was kind of talked about in the original Flow Basics course, but I just want to review it quickly. Basically, the compensation will figure out the mean or median of the positive population and the negative population, and it is compensated when those 2 are aligned. So we want to make sure that we can draw a straight line between the positive and negative populations. So in order to calculate that properly, we need to follow a set of rules for our controls. So 4 rules that I'll go over. The first one is the positive population must be as bright or brighter than the multicolor sample. So I have 2 plots here. Let's imagine that this top plot is our single stain control and the bottom plot is our sample. This would allow for proper compensation because our single stain control is brighter than our sample. But if we had it the other way around, if this bottom plot was our single stain control and the top plot was our actual sample, the compensation would not be calculated properly. It violates the rule. We have a subset of cells within our sample that is brighter than our control, so this subset will not be compensated properly. Rule number 2 is that your control needs to have exactly the same fluorophore as the one in the sample. So hopefully you will just use the exact same antibody that you stained your sample to stain your single stain controls. However, there are people that decide for whatever reason to not follow this rule, and they end up with issues. Now, the most obvious shortcut that people take is if they're trying to compensate GFP and they decide that it's too difficult for them to get a single stain GFP control, and so they'll use FTSE instead, and this is what happens here. So here we have our FTSE single stain control, and it's been compensated on the FTSE. Looks fantastic, but if we actually apply this same compensation to a GFP single stain, you can see how terrible that looks. It's very undercompensated. So please do not use FITSI to compensate GFP. You will end up with compensation errors. Your data won't really look too great. So another scenario where you definitely want to make sure you do this is if you're using tandem dyes, so anything with the dash in it like PE size 7, APC size 7, you definitely want to make sure that you use the exact same antibody. Rule 3. Ideally, you want to have a positive and negative population in your control. Now, this is more critical if you're trying to manually compensate. You do have a workaround for this if you are using a auto compensation method. So you can see here, if you only have a positive population and you lack a negative population, then it's kind of hard to figure out where the two align because there's only one population. If you're using an auto-compensation method, then you can utilize a universal negative, which means that the negative population will be pulled from the unstained tube. And so the computer will be able to calculate that easily, but if you're trying to do it manually, then it's just very challenging to eyeball yourself. Now, the note is if you're using universal negative, so if you're pulling the negative population from the unstained tube, you need to make sure that the two populations have matching autofluorescence properties. So if you have a PE control and a FITSI control, you are allowed to put PE on compensation beads and FITSI on cells within the same experiment. It's perfectly allowed to mix and match cells and beads or different types of cells with different types of autofluorescence properties. But for each fluorophore within that control, you need to have matching properties. So if the PE positive population is in compensation beads, then the PE negative population needs to be compensation beads. If you put the FITSI positive population on cells, then the FITSI negative population cannot be on compensation beads because cells and beads don't have the same autofluorescence properties. You therefore need your FITSI negative population to be on cells. So within fluorophore, they need to match, but within an experiment, you can mix and match for, half your controls can be on compensation beads and half of them can be on cells. That's fine. For the last rule, you need to have sufficient events for both your positive and negative populations. If you are using a autocompensation method, then oftentimes there is a cutoff. It depends on which method you're using. It's usually probably somewhere around 100 to 200 events in the positive gate and the same number in the negative gate. But when you're running it, you can just sort of eyeball it. So you can see here I have a tube where I ran 5,000 events versus the same tube that I ran 20,000 events. And you can see, looking at the positive population, in this tube there aren't very many cells. It's kind of hard to distinguish that positive population, but if we run more events, we can see the positive events much more clearly. So as you're running, take a look at the events that you're running. Make sure that you can visibly see a positive population. If you can't, then you're going to have to keep on running until you see a reasonably sized population. Now, I've been mentioning compensation beads, and I do want to go over the pros and cons of using cells versus compensation beads. So cells usually will be the least problematic in calculating compensation because hopefully they're exactly the same as your cells in your sample, and therefore it's going to give you the best calculation. It also is nice because you don't have to buy anything else. If you use compensation beads, that's something you need to purchase, so an added cost. The cons, however, are if you don't have enough cells or if your antigen is poorly expressed, very rare, then it may be a little bit challenging to use cells because you're not getting a very good signal with an antigen. In that case, you might want to switch over to compensation beads because they just bind the isotype of the antibody. So if you have an IgG antibody, it binds the beads. It gives you a really nice, clear, positive, negative population. It's very abundant, so you don't have to worry about rare markers. It doesn't use up your cells, so you can save those for your actual sample. However, beads are hard to use if you're using dyes or fluorescent proteins, although there is a GFP bead available. I have not really used it, so you're welcome to try it out. And there are times when fluorophores actually don't interact with compensation beads very well, and so even if the beads look like they've been accurately compensated, the cells are a little bit off because of a sort of odd fluorophore interaction with the beads. Now, not all the beads are exactly the same, so some of them bind to different isotypes. Some of them have negative beads mixed in the tube or in a separate tube. The ones that I mentioned that have trouble binding to the beads, or they bind slightly differently and might give you results that are slightly off, are the fluorophores that are made with polymers, which are your brilliant violet dyes, your brilliant UV dyes, and your brilliant blue dyes. So just kind of keep that in mind. And here are the links to the Thermo Fisher and BD beads, which are probably the most popular. I actually did hear that Thermo Fisher came out with some new beads that are a little bit better with these polymer fluorophores, so keep an eye out for those. But it's really up to you if you want to use compensation beads or not. Some people find them to be easier, some people find them to be more difficult. Okay, so moving on to the gating controls, which are useful for analysis. The main gating control, the gold standard, is your fluorescence -1 control, also known as your FMO. And these are basically the opposite of your single stains. They contain all of the fluorophores in the panel except for one fluorophore. And usually the naming is, if you have a PE FMO, that means that it contains all fluorophores in your panel except for PE. And these are utilized to determine what is positive and what is negative. Now, the reason why we utilize an FMO control instead of an unstained control has to do with spillover spreading error, which is a little bit of a more complicated topic. But basically, once you add other fluorophores into the tube, they might cause a little bit of error, which makes the FMO a little bit brighter than the unstained control. And so really you should be drawing your gate based on the FMO control to account for the other fluorophores in the panel. Now here's another example of how you might use FMOs to draw gates. So if we have two colors plotted against each other and we have two FMOs, we apply these gates to the FMOs and then apply the exact same gate to our full stain sample, and you can see now we can determine what is positive and what is negative. Also note that with the FMOs, you have almost all the antibodies in the tube, which means that you can actually gate on a specific subset of cells. So, if you want to use these 4 markers to gate on regulatory T cells using your ICOS FMO, then you can compare the ICOS FMO to the full stain sample to look at ICOS expression. Now, when would you want to use these controls? Again, ideally you want all of them, but oftentimes that's hard to do. So if you don't want to run an FMO control for every single color in your panel, the first place to cut them is the markers that have primary expression. So the on-off pattern, so here's the histogram of CD3 here, it's so distinct from the negative population that you don't really need a control to help you draw a gate on that. However, if you compare that to CD45RA, which is a secondary antigen, it has that sort of continuum or smeary pattern, then you would benefit from an FMO to determine what is CD45RA positive versus negative. And then again, the FMO has to do with the spillover spreading error, which becomes more of an issue as panels get larger. So the larger the panel, the more important the FMOs are. For really small panels, you might not need an FMO as much. Pipetting FMOs can get a bit challenging. This is probably the number one reason why people choose to skip them, is because they're a bit tedious to pipette. You have to pipette every single antibody into every single tube. Again, don't forget that you should not be pipetting small volumes. Please dilute your antibodies before you pipette them into your FMOs. And then I created a little animation video to show my process for how I pipette my FMOs, because it can get a little bit complicated and easy to mess up. So this is my method for not screwing up your FMOs. Okay, moving on to your staining controls. Now, unlike the first two groups, the single stains and the gating controls, these controls are things you need to be thinking about for every single experiment. Every time you get on the instrument with samples, you should be thinking about using single stains and gating controls. However, the staining controls are for setting up your experiment, setting up the staining protocol, so you don't need to worry about the staining controls for every single experiment in the vast majority of flow experiments. These will help you determine if there are issues with your staining, and once you sort out those issues, then you don't need to run these controls anymore. There are two controls that will help you figure out background staining issues. The first one is an isotype control. Now, I do want to point out that in the past, isotype controls were used to gate samples. However, once FMOs were discovered, it's determined that FMOs do a much better job at gating samples, whereas isotype controls do not do as good of a job and are better used for identifying non-specific staining. So you might find some people still pushing the isotype controls, but that is an old way of thinking for gating. Isotype controls should really only be used for assessing non-specific staining, not gating. Okay, so isotype controls, if you don't know what those are, it's a separate antibody. And if we think back about the structure of antibodies, we have a constant region, so this is the isotype. For example, IgG is pretty common. And then we also have two regions here, which are the variable regions, and this is the part that is specific to your marker of interest, or the epitope on the cell. So your isotype control will be the same constant region as the antibody that you're using to actually run your experiment, but it will not have this same variable region, so it shouldn't be specifically binding to your sample. That means that if you find that your isotype control binds to your cells, then that would mean that some other part of the antibody itself is binding to your cells non-specifically and not actually binding to the actual marker of interest. Then the second control is called an isoclonic control. So again, this is a different antibody. It is the same exact antibody as the one in your sample, except for there's no fluorophore conjugated. So the isotype is same antibody isotype, same fluorophore, but different variable region. Isoclonic control is the same variable region, same constant region, but no fluorophore conjugated. And what you would do in this experiment is mix the unconjugated antibody with the conjugated antibody and see if that blocks the binding. So you can either mix them together at the same step, or you can stain with the unstained or unconjugated antibody first and then follow it up with the conjugated antibody. So here's what your staining would normally look like in your sample, but if you include your isoclonic control, then that prevents this antibody from binding, and hopefully it should look like there's no fluorophore staining. However, if you are finding that your isoclonic control has staining, then that means that perhaps there's some free fluorophore in the tube that is binding to the cells, or some other mechanism where the isoclonic control is not inhibiting the specific binding of the cell. So if you are finding nonspecific staining in your samples and you're using these two controls, then what you need to do is go back to the blocking step and optimize that. So we talked about that in the first video mostly of this series. So you might want to think about playing around with that blocking step, or if you're having trouble with that, you could also try using a different clone of antibody altogether. Okay, and finally we have a few other controls. The unstained control, the main purpose of this is to identify if there's autofluorescence in your sample. It is not entirely required to bring an unstained control for the most part, but there are a few conditions where the unstained control is absolutely required. The main scenarios where you would have to bring an unstained control is if you're using an autocompensation method. Those computer methods require an unstained control. If you're using the Cytec Aurora, the spectral cytometer, then you must bring an unstained control. And if you have a single stain that lacks a negative population, then you should really be bringing an unstained control. Remember, it has to match in autofluorescence properties. So I would strongly recommend bringing an unstained control. They can be useful, but there are times when you can get by without it. The main purpose I like to use my unstained control for is setting up the forward and side scatter on the instrument, because then I don't have to waste any sort of sample, especially if I have a different cell type for my controls. The unstained is usually going to be the same as your sample, although if you're using it as a negative or a universal negative, then it should be the same as the cells in your control. The experimental controls, this is really up to you. It's very specific to your experiment, but I just kind of wanted to remind you that you might need to bring some controls based on your actual individual experiment. So you might need unstimulated versus stimulated cells, treated versus untreated cells, wild type versus knockout cells, and in determining which cells you need to use for your controls, that's kind of something that you'll have to figure out for your individual experiment. Or sometimes what people will do is they will mix their cells. So they'll create their FMOs or their single stain controls by mixing their unstimulated and stimulated cells. That's one option that you might consider. And finally, this isn't really a control, but since this is kind of on the topic of experimental design, you might need to think about absolute cell counts if that's something that is useful for your experiment. Now, in order to get cell counts, there's two ways about that. The first way to get absolute cell counts is to add counting beads to your sample. You would have to purchase those, and you can get that method to work on pretty much any instrument in our facility. But you might consider utilizing instruments that actually have a volumetric counter built in. And those are the Thermo Fisher Attune and the Cytec Aurora. Those have a volumetric counter, so you wouldn't need to use counting beads for those instruments, although if you really wanted to add counting beads, you could. Also, one last note, if you are running PBMCs or human blood samples, it is common to do a count per microliter of whole blood. So you might want to think about that ahead of time and figure out, before you process your samples, getting a count from the blood. Now, you're probably starting to realize that there are quite a few controls, and that means you often end up with more controls than samples. So here's an example of a 6-color experiment. We have 6 single stain controls, we have 6 FMOs, we have 1 unstained, and then 6 tubes for samples. So we have 3 unstimulated and 3 stimulated. So that means we have 13 tubes for our controls and 6 tubes for our samples. This, unfortunately, is quite common for a flow cytometry experiment, which means you end up with quite a few tubes. And when you have so many tubes, you start to worry about how many cells you use for each of those tubes. So I also want to talk about how you might go about dividing your precious sample between all of these tubes in your experiment. I do want to remind you that what we talked about in my second video of this series is that changing the cell number often doesn't have a huge impact on the staining intensity of the cells. So you probably have a little bit of flexibility with the cell number, which means you can cut back on the cell number for your cells, for your controls. And again, I do have a blog post that goes over all the considerations you would want to make in terms of adjusting cell number. But for your compensation controls, your single stain controls, usually these controls can survive with fewer cells in them than your samples. Again, you just need to follow those 4 rules. So you need to make sure that you have enough cells to see a positive population or a negative population, if that's what you're looking for. And the staining needs to be as bright or brighter than the multicolor sample. So the factors, if you think back about how the staining protocol affects your results, you might end up with a dimmer control if you have fewer cells. Again, it's probably less likely, but it's still a possibility. If you reduce the number of cells, you might get reduced intensity of the fluorophore, or if you screw up your pipetting and you accidentally pipette a smaller volume than you intended to, which is the problem with pipetting small volumes, you might end up with a dimmer fluorophore on your control than the multicolor sample, and then you will run into compensation errors. So just keep those in mind. For FMO controls, remember we talked about how you might be able to cut out a few of those controls from your experiment if they have the primary antigen on-off type. And then depending on the marker, ideally you probably want to try to keep the FMO controls at the similar cell number as your samples, but it kind of depends on the marker itself. So if you have CD45, which is very abundant, it's expressed on most of the immune cells, then you probably don't need a ton of cells in that FMO control. But if you're planning on looking at a marker on a really rare subset of cells, then you probably want to try to keep the cell number in that FMO control as close to your sample as possible. So to summarize the last 4 videos, the steps to set up your experiment. You're going to want to determine the goals of your experiment and keep those in mind through all of those steps. Those are going to help you choose your markers, design your panel. Remember that you need to figure out which instrument you're running on, plan your panel for the instrument. Then I strongly recommend titrating your antibodies, and if you have the time, testing out the full panel of fully titrated, optimized protocol on your actual tissue of interest. Ideally, if you can't do it on your actual tissue, at least do it on a different tissue. Test out the panel, make sure you can identify the cells that you're looking to identify. Then also what we discussed in this video, figuring out how many cells do you actually need to stain, which controls do you need to stain, how many cells do you need for those controls. And then once you've figured out all of those steps, you are ready to run your experiment. And stay tuned for the next video where I will be giving you a few tips on how to set up the instrument, and I'll be discussing compensation further.